Hi, my name is Nathan. I'm a fifth year graduate student here at the University of Arizona. I work in the research lab of Dr. Oliver Monti, and then I also have a position as an RA at the Keck Center for Nano Imaging. The Keck Center for Nano Imaging is a facility in our chemistry department here on campus that is a multi-user facility. And basically, we have several different instruments to analyze samples, mostly at the nanoscale, as the name implies. And we work with people from all departments on campus, so not just chemistry, but also physics and optical sciences and chemical engineering and just a lot of different people. And we train graduate students and undergrads and postdocs to use our instruments so that they can come in and use them on their own time to do their research. So the primary instruments we have in Keck that I help to run are we have a scanning electron microscope, which you can see behind me in this fake Zoom background. This uses a beam of electrons instead of a beam of photons to take images of samples like a light microscope would. And the electrons allow us to take images down to a resolution of about 100 nanometers, which is much smaller than you'd be able to get with a regular light microscope. So that's pretty cool. We also have two atomic force microscopes, or AFMs. And these use a very sharp tip that you can drag across a surface, and it will follow the topology of your surface. And so people use this pretty commonly to measure surface roughnesses, again, down to, in this case, the nanometer scale, or step edges, kind of the height if you have your film that you've grown on something, for instance. Then we also have an IR spectrometer, infrared spectrometer, that people can use in a few different modes to take IR spectra of their samples. And then finally, we have pretty recently a thermogravimetric analyzer, or TGA, which is basically just a very fancy oven and a very fancy balance or scale. And so it allows you to weigh your sample as you're heating it up. And see it lose mass as it thermally degrades. And so people use that to measure thermal stability of compounds, or kind of if there's some component of your compound that all goes away at some point and you want to know what percentage of your sample is made up of, water or something, say. So those are, there's a few other instruments too, but those are the main ones that I help to train people on. I think the coolest sample that I've helped someone work with so far is there's a group, I believe in biology, who's studying mosquitoes. And so they had these mosquito eggs that were hatching into mosquito larvae. And they were able to basically, I think they like froze them or somehow like basically killed all of the mosquito larvae like in the instant that they were starting to emerge from the eggs. And so they had these frozen in time, partially emerged mosquito larvae that we were looking at on the SEM. And they were just some really cool and gnarly pictures of these half emerged mosquito larvae that we could see in really exquisite detail on the scanning electron microscope. And it was really cool to look at it. It's nothing like what I do, so it was really cool to see that other side of different types of research that people do.